Thank you, Professor Daniel Tommer. So next speaker is Professor Chung Lei. So Professor Chung Lei from Xijin Hospital in Xi'an. So she is the Deputy Chief of Anesthesia Department. So welcome, Professor Chung Lei. Give us the lecture is as the topic is using an unicorn MAC as the indicator of anesthetic potential for validated anesthetic protocol, Profen and OPs. Welcome, Professor Chung Lei.
Thank you. Thank you for the introduction. Thank you, Fiona, for the invitation for me have this opportunity to give this talk about using an equivalent MAC as an indicator of anesthetics potency of the violate anesthetic propofol and opioids, and representative of the our research group, leading by Professor Dr. Dong Hailong, and I would like to report some of the recent work here of our research team. So first of all, I would like to share with you the future of anesthesia. Maybe a lot of you have attended the previous section this morning, that the technology vision of the anesthesia in the 2040. So due to the development of the drugs and the monitors and equipments, and we have greatly improved the patient safety. And future, the ideal anesthesia would be like precision and for the personalized optimizing, and with very improved the patient outcome for after the surgery or anesthesia perioperative management of the patient. So we all know that the general anesthesia is a drug-induced reversible state of the unconsciousness, immobility, and the blunting of the adrenalic response. So for the immobility and the BAR, the bar, is straightforward to monitor. Like we can use MAC or MAC bar to indicate the probability of the patient response to this noxious stimulus. But some kind of it's complex for us, for our anesthesiologists, during the management of the patient intraoperatively. It's hard to monitor if the patient, the status of the consciousness. As we all know, if the patient is well, like consciousness, we all text the intraoperative awareness used post-operatively, like questionnaire, to ask the patient if they can recall anything during the surgery. So, but we all know the famous isolated forearm test, where the patient was used a tourniquet to isolate the forearm and make the arm unparalyzed, and used during the surgery and during the in the anesthetic status. They ask the patient to the verbal command, ask the patient to like the two response for the verbal command. And there's the results shows that with the processed EEG or BIS, the non-responders and the responders are there's no good separation of the BIS can be like to separate the non-responders and the responders. So it may be indicate that the intraoperative unconsciousness states is kind of complex to indicate, and we maybe cannot use the consciousness, as we can only say that the patient was no response to the noxious stimulus, or we can say there's no explicit recall of the steps during the surgery. And the other thing is that we all know that we can use the MAC to monitor all three components of the anesthesia that we mentioned: unconsciousness, immobility, and the blunting of the energetic responses. And we know that the probabilistic nature of the MAC requires that we need to get at least or almost 100% and 95% or 85% to the component of the consciousness, immobility, and blunting of an adrenalic response, respectively. But we can see from this graph that they use the fraction MAC. They are quite separate, these three components. But this MAC was indicate the volatile anesthetics alone. But we all know that during the surgery, we cannot it's a combined anesthetics we are using. So we all know that when combination use of opioids and volatile anesthetics, they have different synergetic effects. As for the consciousness, if combining use the volatile anesthetics and the fentanyl, the max curve shift about 15%. But for the immobility, it's about 58% and 75% for the blunting of energetic response. So if they all taken together, we can all of the three curves are left shift, but they can be cause the synergistic effects are different. So we can see that after combination use of the opioids or fentanyl, the three curves are getting together. And if at this time 0.7 fraction mark will be ensure the three clinical goals can be achieved. So as also indicated by this research published in BJA, that bees do not work well when combined used with high-dose opioids. So what can we do? Because clinically, when we using the FMAC to guiding us or titrate the optimal anesthesia or anesthetics, we need to keep in mind that the nature of MAC was probable leak. And we need to know that there is getting obtained from the volatile anesthetics, and there's no information about combination of use different anesthetics, and there's the drug interaction, and also there's a heterosis. That means there's time lag, especially during the unstable status, like we are doing anesthetic induction and during the emergence, and during when we are changing the dose of anesthetics, because there is time lag from the plasma concentration of the anesthetics to the target tissue, in the brain. So there's commercialized monitored device or workstations to address this problem. Like we all know, the Mac brain, they use the separate Mac brain for indicate to address the hysteresis adjusted or corrected Mac. And also the Smart Plot and the Navigator by the two different companies, they integrate the different drug interactions, especially intravenous drugs interactions. They convert the response surface model into an isoborogram. But the problem is it's hard for the clinician to rate at this time. Use this complex pharmacokinetic model and to rate and to interpret and to instant guiding for the titration of anesthetics. So most of these models are used the multiple anesthetic drugs, and clinically most of us are working by our experience. There's no precise index or guiding for us to know the exact the drug interventions of if they are synergistic or addictive. So using the same concept of the MAC as we used for guiding us to adjust the volatile anesthetics. If there is a possible that we can generate the equivalent MAC guiding us for when we are using the intravenous anesthetics, that will be improved the clinical patient safety or the individualized precision of when we do the patient management. So most of the combined effect of multiple drugs are reflected by the response surface here, and after that they will convert to the isoborogram, and the mostly used is the hierarchical model, and commonly used the PK and PT model for anesthetic drugs here. So this combining drugs use the PK and PT model by the surface response surface, and we can convert that into the EMAC that use the same concept or algorithm with the same smart plot or navigator. But here the Mindray generate the equivalent MAC here. They transferred into an index that will be like similar to the MAC we are routinely used clinically. That will be easier for anesthesiologists to understand, and for very simple instant adjustment of the anesthesia. So here is the advantage of this anesthesia site is that we can incorporate all of the parameters we need to do the patient management during the intraoperatively or postoperatively. Here it was incorporated in a single workstation. We can have centralized control and the combined drug effect here, we said, convert into the equivalent MAC value. And here in the right side of the monitoring, the screen, you can see there is different, the intravenous, we use the TCI model for the intravenous anesthetics. And here we can see that the EMAC was showed in the upside of the screen. So here is the algorithm for calculate the equivalent MAC for the propofol or other opioids. So first we calculate the total potency and then probability of no response to noxious stimulus, and then convert to the EMAC calculation. And this is use the hierarchical model and convert to isobologram. And here, after that, and we used in the clinical practice, we analyzed 53 patients and to see if we can use this equivalent MAC to indicate the anesthetic potency of inhalated anesthetics, propofol, and opioids. This is the process. And here we can see that we chose two combinations, that is sevoflurane combined with remifentanil and the propofol combined with remifentanil. And after that, we can see that we have 60 data points during the anesthesia introduction and emergence, and the 300 data points during the maintenance. So this is the characteristics of the 53 patients. And here we can see that the prediction probabilities of the EMAC for and the tolerance to skin incision, the probability and the beats are they perform as quite differently. For the immobility, blunting heart rate, and blunting blood pressure, the prediction probability was significantly higher for the EMAC when compared to the beats. And this is in accordance with the previous study published. It demonstrated that the verification of the accuracy of the combined drug effect model, that's the equivalent MAC was outperformed the beats. And we can see here there's a concordance between the EMAC and the beats during the propofol combined remifentanil and sevoflurane combined remifentanil anesthesia. We can see that the curve during the whole intraoperative anesthesia is quite similar and stable. And here is the distribution of the MAC. We can see that from the whole cohort or the two separate anesthesia, the combinations, we can see that the distribution from the 1.3 to 2 or 1.3 to 2.6 of the EMAC was about 52.7 and 60.4 and 44.4%, and the other was around 72, 75, and about 70 respectively. So we can see that for the intravenous anesthetics combination, the EMAC distribution was narrowed when compared to the sevoflurane combined with remifentanil. And this is the EMAC awake 50. That means like 50% of the patient will be awake at this EMAC value. It's around, the mean value is about 0.03, which is pretty much like the MAC we used for the volatile anesthetics. And again, this is show the screen of the anesthesia site. And we can see that the take-home message, we can see that the EMAC can guiding us for the drug titration and drug administration, and it's easier for clinical training because we don't need to rate the isosobrogram of the PK/PD model for the drug combinations. Just convert to the one single index. And here, I'm not sure if you can. There's a video here. I'm not sure if you can. So we can see this is a video used in hospital because all the parameters are integrated. As this morning it says, there is no matter how many parameters makes sense is we use the right parameters at the right time with the right indication, and we can use it rightly. So to summarize, the anesthesia status integrate its assessment of the prediction was quite incorporate all of the parameters together, and we can achieve a remote control of the infusion pump. And here, like the depths, the future, the perspective, the future of the depths of the anesthesia and the nociception monitoring, we can see that the patient-centered, we can do like, we can incorporate all the things together and achieve precision patient monitoring and finally improved patient safety, and use the multiple devices to incorporate into all-in-one station and with wireless in the OR. So that's what in the future. So first, we just a little bit step forward at this path. We are reaching the future. And here is some cases. We can see that with the like control bar in the screen, we can just control the infusion pump. There's no need that you can there's no need to touch on the infusion pump. And here for the neurosurgical procedures, we can see that the BIS monitoring will be interfered by the electrosurgical knife, but the EMAC was achieved like continuous monitoring of the. And here, I think our future efforts would be the updates and iterations of the monitor, like EMAC. We can make it more precise and put more data and like use the artificial intelligence to revise or update the algorithm of the EMAC we get, and to compare those with the similar products. And more drug combination, here we just use the propofol and remifentanil, and maybe the other drugs like ketamine, dexmedetomidine, or etomidate should be like incorporated in these PK/PD models to generate accurate EMAC for us and more accurate prediction for the patient. So the final target was improved patient safety. So here I think it's the bottom line to get the titration or individualized anesthesia. Finally, we want to like use these techniques to predict. So, and we can like to react in advance. So finally we can like reduce the post-operative complications and comorbidities, and that's what we like retrieved, like we target for. And so the future anesthesia patient monitoring are here, like the prevailing, like we use the different technologies, and we use the future technique will help us to make everything more precise. We can react in advance, and we can finally improve patient safety. And I think the future anesthesia workstation will be closed-looped, integrated, and in precise prediction and titration for every individual patient. And that's all. Thank you for your attention.